We're going to get everybody settled here. I see people are piling in. Grab a seat in the front. Oh, that's right. You all are at the front because you are on your laptops today. Um, Welcome. And we've got our special session. Now, in addition to working on you know, projecting our own research and research output, we also like to try to find links to other research programs. And so it's my delight today to invite the team from the Water Reuse Consortium. And it's sort of like the Partridge Family. You know, they're a group of individual singers in different parts of the bus, but they're all working together to advance water reuse. And before we begin, I just want to go through a couple housekeeping items, as we always do. These meetings are recorded, and so you'll be able to get a link of this, and then if you have other colleagues in your organization, you can share that link with them. We'd like you to stay muted during the duration of the presentation, but when questions come to you, please put them in the chat. And then at the end, in the last 10 or 15 minutes, I'm going to actually unmute you. So if you've written a question, I'll invite you to speak your question aloud so you can talk to our team directly. And also, if you aren't already in our LinkedIn family, do link us in, because that is how we are advising and updating everybody on the schedule of webinars. So seriously, it's right here. You can shoot this QR code and you'll get all the updates, including updates and information about our upcoming funding opportunity, which is coming soon. I know I said that to you last week, but it's still coming soon. And this will be a great opportunity in the area of industrial water reuse piloting. Now, if you are curious about some of our past webinars and other presentations, we have almost 100 recorded in our YouTube channel. So you can go back there, even back several years. And even still, some of this information is frontier research in advanced water treatment and desalination. We always now have to say this, that this is NAWI, this is not the Department of Energy, and our member laboratories, we are speaking as individuals. And we also want to tell you that our next month's webinar is a great one. This is on June 17th, 2026. This is Professor Yorm Cohen and the team from UCLA and Project 510. He'll be talking about the project we call Waymo for Water. A very successful demonstration of advanced water treatment and autonomous treatment. These are little water treatment systems that an operator needs to visit roughly once a month, and they've been running effectively. For those of you who subscribed to Maven's Notebook, the California Water News feed, this was the featured story yesterday. So check out that. That's a great result. And by the way, Nick, this stuff would be great for forward deployed forces or small remote bases. So, you know, there's lots of places for us to play together. And that's kind of what we're doing today. So I want to welcome the team from the Water Reuse Consortium, Amy Childress and Adam Smith from USC, Andrea Achille and Carrie Higginbottom from University of Arizona, Sage Hibble from University of Nevada, Reno, and Nick, welcome from the core. And I'm at this point going to pass this on to Amy. Amy, thank you so much for gathering your community. We're looking forward to learning about not just the sort of threads of research that you all are doing, but some of the broader things that you all are thinking about how we advance water reuse in today's arid and parched world. So with that, I'm going to stop sharing and hand it over to you.
All right,
Have you got it?
wonderful.
Good.
Good. Okay, you see my screen?
We do.
Wonderful. Okay, so thank you for having us, Peter.
You can put it in projection mode.
Oh, sorry.
Yep.
There
No
we go. I haven't done this in a little while. Continue to share. Okay.
Okay, looks great.
Okay, wonderful. Good. So I'm going to give a brief overview of the Water Reuse Consortium and then pass it to Nick, who will talk about our role in military water security and resilience. We'll have 3 research snapshots, and then we're trying to save 15 to 20 minutes in the end for open discussion. Peter's already introduced us. We are the leads from these institutions, but there are many other people at our institutions who support this research. And our goals are ensuring water security and resilience. We do this by partnering with industry, government agencies, and communities and we are delivering technology, education, and communication tools. And we'll talk about those. But at the core of what we're doing is our engineering and science research, but also very important being academic institutions, education, communication, and tech transfer. So if you're wondering where we started, we actually started in this defense space. So you'll see that as an underlying theme throughout defense and also the duality of military civilian reuse systems. But our first system, which was more than a decade ago, was specifically for expeditionary missions. So it's potable reuse on expeditionary missions. And essentially, and you can see early days of Andrea and Sage here. But of course, the goal is if you've got these forward operating bases with hundreds to thousands of personnel, the greatest risk incurred by these actually is from the water and fuel convoys going there, the wastewater leaving there. So if you could have a system that reclaims wastewater for reuse on site, and especially one that does it with less energy, you could save the risk of these water and fuel convoys. So that was our starting place. We took what we learned from that and moved it forward, leveraged it into a system. It's a containerized potable reuse system that was first tested in a municipal application and then moved to Fort Huachuca. And again, you can see team members here, me and Andrea, and here's Nick. And the key thing of these installations is they have a mandate for self-sufficiency. So they have to operate for at least 14 days of water and energy inside the installation fence. And of course, water reuse is a key way that can be achieved. But what I'm going to do in the next slides, I'm going to talk a little bit about our training, education, outreach, and communication, and then pass it off to discuss the heart of what we do, which is the research. Of course, as academics, part of our key thought here is workforce development and the training of students who understand what potable reuse, in particular, potable reuse is. But we're not just trying to teach them the basics of potable reuse. We want to facilitate industry and academic exchange so that we're sure that the research gaps that we're targeting are ones that are of interest in the real world. And so you can see a lot of the guest speakers we've had, and we have often a strong contingency from the Trussell team as well. We've only offered this twice so far. It's coming again this fall. We also offer hands-on training through an advanced water purification workshop. This is not just hands-on on equipment, but also software simulations. And this training, in fact, has been offered to AICHE, NAWI, Water Tap, and West Point cadets most recently. We've had we were able to advertise this hands-on training in our recent call for utility operators. So we had representatives from Metropolitan Water District, LA County SAN, and other locations recognizing this need for operators, and not just any operators that are highly trained in these systems of the future. And so that's a reminder of that. But of course, also, we realize that although we think everybody's on board with potable reuse, that may not be the case. And so there's been some great work done measuring the public perception of risk and specifically targeting gaps and opportunities on how to bring the public along with us. And a key reason that we want to do, or a key way of doing that, is we've hired, we work with an Emmy award-winning filmmaker who has created a documentary series. These are short documentaries. The first features Dan McCurry in the past and the future water supply to Los Angeles. The second features Carrie Hickenbottom, shared water, because of course we are 3 states in the lower Colorado Basin. The third, that was me again, is the Water Masters, and that's Sage Heibel and Eric Marchand on the Lake Tahoe Pyramid Lake system. And then the fourth, which is coming soon, Guardians of the River, features ERDIC and all of their work on the Mississippi River. And very important, if you want to watch these, don't think you need to clear a whole weekend. They're only about 30 minutes each, so you could do it in one sitting. And of course, we have a code in the top to scan if you're interested. Again, our goal is to try to instill confidence and trust of the public. And then also we've kind of saw a hole in connecting the potable water reuse community, especially if we consider that Army installations exist across all the 50 states, which means they are subject to the regulations in all of those 50 states. There needs to be better communication and exchange, particularly with regard to regulations, but other things as well. So this is the potable water reuse report. Series 1 was on regulations, Series 2 pathogen crediting, and now we're in Series 3, issue 2 on chemicals is coming out soon. This is in collaboration with the Trussell team. They've been wonderful collaborators at the table with us. And I also have to do a shout out to our advisory panel, Sharon Napier, Dawn, who is now Nick Josephic, and Jeff Mosier and Tom Pankratz. Dawn Morrison, by the way, she started with us. Okay, and then what I want to do is hand it over to Nick to talk about our role in military water security and resilience.
Yeah, thank you so much, Amy. I really appreciate you all letting me be a part of this consortium and part of this presentation. This is great. Many of you are probably wondering, why is the DOW, the DoD, concerned about water resilience, water reuse? Well, typically it's because we have very specific missions that we have to accomplish, and usually those missions include humans, and typically humans like water. So we cannot accomplish our missions without having the right supply of water, having water security, having it reliability, having our systems resilient so that we can do it. It is a weak point. We're looking at both water resiliency and at both the fixed installation and in the forward operating bases. So at the fixed installations, they're like small cities. They have to operate every day. And during an emergency, they're a point where everyone is going to converge. So we have to have reliable, safe water. And we could store a heck of a lot of it, but water reuse is one way that we can incorporate that in there. So all of our day-to-day activities, we have wastewater treatment plants on our fixed installations. We have to figure out how do we optimize that? How do we increase the resiliency? And by implementing new technologies, retraining humans on that installation, and then also redoing rules and laws to allow us to do certain things. So those are some of the aspects on the fixed installations. And as you all know, as Amy mentioned, that we have this requirement that we have to be self-sufficient for up to 14 days in an emergency. That's a heck of a lot of water to store. Reusing water will help us meet those goals. Coming down the pipeline, we have data centers. They are looking at putting data centers on military installations. We have the land, we have the needs, we have the humans. Do we have enough water? How is that going to affect both the installation and the areas around it? So those are some of the things we're looking at. Then if we go over to the forward operating base, again, we have missions there. We don't know what water supplies will be available. We know that supply convoys are an area that could be a target, and anything we can do to reduce those convoys will increase the security for our soldiers and potentially help support our mission. So we're doing research in both of those areas, trying to implement things for the DOW. But how do we do that? We can't do that without you. So we do it through partnerships like this with universities. We do it with other federal agencies, and we do it with the public sector. There's no way for us to do the entire research by ourselves. The only way we can do it is through these partnerships. and that's the real strength in pushing these technologies forward. So the other partners, they're each going to tell some very specific projects and themes that we're working on in this Water Reuse Consortium, but it is really just scratching the surface of the breadth that we've been able to accomplish with this Water Reuse Consortium. And so we hope that we can just give you a snapshot with these next few speakers of just a few spaces, few small parts of the really impactful research that they're doing. So I will pass to the next gen biotech.
Awesome. Thanks, Nick. All right, Adam, you're up.
All right. Thanks so much, everyone. I'm excited to share the first research snapshot from the consortium. Amy, you can go on to the next slide, please. Okay, so some of the work that we do in the consortium is focusing on what I call next generation biotechnologies or wastewater processes that are compatible in potable reuse schemes. And so on the left of the slide, I'm just showing a simple direct potable reuse scheme where wastewater treatment is our first line of defense between our wastewater and, of course, our advanced water purification facilities. And although these wastewater treatment plants don't always get the spotlight in potable reuse conversations, really innovations are needed to enhance their compatibility with downstream advanced water purification, but also improve energy requirements, reduce carbon needs for nutrient removal, and then also deal with the control of micropollutants because they really represent our first line of defense. So what next generation biotechnologies, and this is not an exhaustive list, but this is some of the ones that we're interested in. So I have 3 schematics shown here on the slide. On the left, we have the anoxic aerobic membrane bioreactors. And these are gaining momentum due to their ability to improve effluent quality relative to conventional activated sludge processes. So this biotechnology provides both carbon and nitrogen removal, and it has built-in ultrafiltration membranes for solid separation. And so what this does is it eliminates the need for that first microfiltration or ultrafiltration step at the advanced water purification facility. In the middle, I have another process, also a membrane bioreactor, but in this case, fully anaerobic. And its benefit is that it biologically converts organic carbon and wastewater into methane-rich biogas, which is a renewable energy source. So it's directly recovering energy from wastewater. It also has built-in micro or ultrafiltration membranes, so it improves our compatibility with advanced water purification. I also want to point out these anaerobic membrane bioreactors generate significantly less biosolids compared to aerobic activated sludge processes. So this is a huge cost savings for disposal. And then finally, on the right, I have a membrane biofilm reactor, which is a little funky in the way that it uses membranes. It relies on a counter-diffusional biofilm for treatment. And so we have microorganisms that grow directly on this hollow fiber membrane, and the hollow fiber membrane is delivering oxygen in a very efficient manner. And so it's reducing our energy requirements. And it also does this at a much lower operational footprint, which is increasingly important as we're retrofitting facilities for reuse applications. And then we also investigate micropollutant fate across these conventional wastewater processes and, of course, these emerging biotechnologies that I just introduced. So for today, I'm just going to focus my discussion on antimicrobial resistance. So we consider this a biological contaminant of concern, and it occurs because of low levels of antibiotics that are present in our waste streams, and then high concentrations of microorganisms that we rely on for treatment. And so this creates conditions that can propagate antimicrobial resistance, resulting in its presence in effluents that can be released to the environment or sent to advanced water purification facilities. But it's also present in biosolids, and that's a major concern when those biosolids are land applied. And so we like to think of antimicrobial resistance as a one health problem where resistance in humans, animals, the environment, and clinical settings is all connected and of concern. So the first study that I want to introduce here briefly is our attempt to understand antimicrobial resistance across conventional activated sludge systems. And so to do this, we partnered with utilities in California, Texas, and North Carolina, representing a cross-section of the United States. And we sampled the influent, the activated sludge, and the effluent from full-scale facilities across 3 consecutive weeks. And we wanted to go beyond simply quantifying resistance genes, which has been done for the past decade or so. We wanted to know which bacteria, especially pathogenic bacteria, are actually hosting these resistance genes. And this can be particularly tricky with antibiotic resistance because a lot of these genes are found on plasmids, which are these small circular strands of DNA that are really easy to spread via horizontal gene transfer between unrelated bacteria. So to determine these host bacteria of resistance, we used a new molecular biology method known as proximity ligation sequencing. And I won't go into the nitty-gritty details of it, but it basically physically links DNA within a single bacterial cell so that with high accuracy, after we do sequencing, we can determine which bacterial cells are hosting which antibiotic resistance genes. Next slide. So to get into some of our results, this figure represents the antibiotic resistance profile across the 3 states we sampled, and that's along the top of the plot. The 3 sample types are along the secondary y-axis, and then the consecutive weeks are along the x-axis. And along the y-axis, the data is represented as relative abundance of resistance genes according to their antibiotic class. And if you just kind of zoom out, I know it's a lot of data to look at once, but if you look across all of these stacked bar plots, really you're seeing a fairly consistent resistance profile, regardless of the state, the sample type, or even the week. So we have this diverse resistance profile that's fairly persistent across treatment. And of the roughly 20 major antibiotic classes in existence that we know of, 16 were detected in our study. So a fairly high number relative to antibiotics that we're aware of. And I do want to point out, though, this data is relative, not absolute abundance, which is the limitation to the sequencing approach we used here. But we do address that in subsequent experiments I'll discuss in a couple slides. I just want to make sure that this doesn't give the wrong message. Our treatment plans do provide removal of resistance genes, and it's typically in the range of 1 to 2 log removal of ARGs from influent to effluent.
And Adam, I'm going to click to the next slide, but we need to wrap.
Oh, okay. So which bacteria are hosting these genes? So we found that only most bacteria, or about 4 families of bacteria, were hosting most of our genes. And here you can start to see the decrease in resistance genes from the influent to the effluent. And so what this work shows us is that we can target specific bacteria via interventions to lower resistance across treatment. And potentially we can do that in some of our emerging treatment processes. And so if you go ahead, Amy, to the next slide, please. And I'll skip this and go to the next one to save time here. So we wanted to study some of these emerging biotechnologies in the lab and specifically look at resistance. And so we did a comparative study with 2 bench scale systems, 1 aerobic membrane bioreactor and 1 anaerobic membrane bioreactor. And we fed these systems with different antibiotics to provide a selective pressure. And we used the same suite of different molecular biology tools to characterize resistance over time. And so on the next slide, so on the left figure, we're comparing the aerobic MBR and the anaerobic MBR over 50 days of operation and total ARGs that we quantified. So this is absolute abundance. And so you can see across the aerobic system, we're seeing a slight increase in resistance over time. That's not something we want to see. And then on the anaerobic system, we're actually seeing a very significant decrease in resistance over time. So that is what we want to see in terms of a technology that limits the propagation of resistance. And then on the right-hand side of the slide, if I have one more second here, we're on the x-axis, we're looking at different antibiotic classes that were observed. And on the y-axis, we're looking at different types of bacteria that hosted that resistance. And it's a heat map and it's kind of complicated, but ultimately we just want fewer number of squares over time, meaning that we're lessening resistance. And we also don't want to see resistance to multiple types of antibiotics in a single type of bacteria. So I've put those in a red dashed rectangle. So that represents multi-drug resistance. We see that is increasing in the aerobic system. And on the flip side, in the anaerobic system, it's actually decreasing. So broadly, this work advances our ability to track resistance in high resolution in biological systems. And we're seeing some advantages when we look at fully anaerobic processes relative to their aerobic counterparts.
Excellent. Thank you for that snapshot, Adam. All right, Andrea.
Hi, yes, happy. Thanks for having me here. Yes, let's switch to the. So if we look at the inland water cycle, if where we need to implement either direct potable reuse or indirect potable reuse, the one main concern is the disposal of the concentrate if we go with an RO-based treatment technology. And if we go more in details with the next slide, please. In the full advanced treatment for potable water reuse, when you have microfiltration, ultrafiltration, reverse osmosis, and then advanced oxidation processes, common challenges include the low recovery of the RO membrane due to scaling and the high RO cleaning needs due to fouling. And so what one of our innovations, yes, next slide, please, for treating the concentrate of the RO, and so to recover more water to dispose less concentrate, we came up with these stream technologies, in which is basically at the heart of these technologies there is this electrolytic cell which produces a base and an acid from our secondary RO concentrate, and then we use the base to remove calcium and magnesium in the FBCR, which is a fluidized bed crystallization reactor. And then we use the acid to produce a ferric coagulant in an ion reactor so that we can remove all the organics. So this is it looks complex, closed loop treatment with aim to be an electrified and decarbonized way to have like acid and base on site and to clean secondary concentrate from RO. So at the core of these technologies, there is this electrolytic cell in which in the anode we produce acid and base. Sorry. Next one, please.
Okay, sorry.
So at the core of these technologies, there is this electrolytic cell, and we would produce acid and alcohol as well in the anode and base in the cathode. People may ask why we went with just a typical chloroalkali cell instead of bipolar membranes. And the reason is because it's way more robust and we can switch the polarity so that we prevent fouling and scaling of the electrolytic cell itself. We tried a lot with the bipolar membrane and it didn't really work. And we can manage the chlorine in our system. So next slide. We started with bench scale results, and we have the concentration on the primary y-axis, and also the TOC concentration on the secondary y-axis. So the RO in the FBCR with the base, we remove virtually all of the magnesium, half of the silica, which is really good, and most of the calcium too, more than 90%. Sodium increases because we introduce the base, and also the TOC remains pretty much unchanged. When after our iron reactor, so after coagulation, we are able to remove about half of the TOC, and as you can see, there is quite a few chlorine because of the production in the electrolytic cell. Although we can manage chlorine with the GAC, and we can also remove further TOC. So these are very encouraging results on the treatment of this RO concentrate. We put that solution and we tested with a bench scale reverse osmosis. Next slide, please. And if we see, this is the normalized flux in the y-axis as function of the duration. This was a concentrating experiments that ran for 2 weeks. So the RO concentrate as is, has a very sharp decline in water flux as expected because scaling and fouling. And as we introduce, coagulation or crystallization and coagulation, we were able to maintain high flux for a very long period. So with those results, next slide, we are now at the West Center building a full pilot of this technology. We can see the electrolytic cell on the left, the fluidized bed crystallizer, and then the ultrafiltration and granular activated carbon and reverse osmosis. And we already have some results for the full-scale pilot scale plant. And next slide. Yes, and this it virtually works as well as the bench scale. So we remove most of the scalants in the FBCR, and then post the ion reactor, we are able to remove substantial TOC, and we have some chlorine, which we believe we will be able to remove with the GSE. And then so for the last slide, we have a very fruitful collaboration with NAWI, with their water tap team. They are helping us to build the digital twin of the process, which will inform on how to operate it at the pilot scale. They are developing new unit models for us or with us for the crystallizer, the ion reactor, and the electrolyzer. And then we use their platform in the Reactoro and water tap to model the full system. So we are very grateful for this collaboration. And I just want to conclude in the next slide with the few key takeaways. So we can have on-site generation of reagents using electrified processes. We expect to have a water production rate of about 90% once we remove scalants and foulants. We can have longer memory lifetime and less cleaning frequencies than other competing technologies. And this is, I believe, it's has the potential to be a universal pretreatment for different types of brines. If you have more scalants, you just use the FBCR part. If you have foulants, you can use just the ion reactor part. And then this was an idea from Jim Farrell from here at the U of A. I want to acknowledge the collaborators, Carrick Hickenbottom, and all the NAWI team and the many students that worked in this project. Thank you.
Super, Andrea. Thank you. Next up is Sage.
All right, thank you, Amy. So this project, or I guess rather series of projects that I'm going to share, are a couple of examples of the utility academic partnerships that the consortium has worked to cultivate. Topic, you can see there, dissolved organic nitrogen is of relevance to any of our utilities or industries across the country that are facing these ultra low nitrogen discharge limits. And really the projects have grown from bench scale studies in the lab that were sponsored by our utility and our government partners to address nitrogen discharge challenges that we face here in Reno. For those of you that don't know, our primary source of drinking water is the beautiful Lake Tahoe. And you can see in the map there that we've got a pretty short transfer through the Truckee River going from Lake Tahoe through Reno. So all of our wastewater discharge ends up back in that Truckee River and ultimately discharges to the Pyramid Lake, which is a terminal lake with no outlet. So its inputs are very strictly regulated. Since those early bench scale studies, the scale of the projects have grown, and shown on the right there is a schematic for the regional indirect potable reuse project that I'll mention at the end of the slides here. So next slide. So as the title suggests, we're going to focus on the removal of dissolved organic nitrogen, or DON. This specific fraction of total nitrogen is going to be classified as the organic component of anything that'll pass through that 0.45 micron filter. So it's going to exclude your nitrates and ammonia. And it's known to be particularly recalcitrant. It's usually whatever's left over at the end. So on the bottom left-hand side of the slide, I've got some examples of DON species that are common in wastewater. They range from the very small molecules, simple molecules such as urea, to aromatics, to large molecules like proteins. And then on the right-hand side, I'm showing some representative HANs and HAMs, which are DON species that are actually nitrogenated disinfection products that can be created during disinfection processes throughout the treatment train. Next slide. So the results that I want to highlight today come from two separate pilot scale studies. So the first one was a enhanced coagulation study, lasted about 11 months at our Truckee Meadows Water Reclamation Facility, or TUMWRF. And in this case, we were looking at 3 different coagulants for DAWN removal. And then the second is a little shorter. It was a 2-month-long study as part of a larger IPR demonstration located at one of our other regional WRFs. And in that case, we were tracking, among other things, both DAWN and dissolved organic carbon throughout an advanced water treatment train. So these are both pilot scale systems. You can see from the photos there that we containerized our systems into the semi-truck trailers, and they had flow rates between 5 and 10 GPM for both sets. Next slide. So as mentioned, nitrogen from our wastewater treatment facilities is going to be strictly regulated here in the Reno area. Most of our effluent ends up in that terminal pyramid lake. So TUMWRF is our largest treatment facility in the region, and it's got some of the strictest nitrogen limits in the country. So we're capped at 500 pounds per day. At 30 MTD, that works out to be about 2 milligrams total nitrogen per liter. And in our case, almost 90% of that nitrogen coming out as effluent is going to be in the form of DAWN. So any of the treatment technologies that we're able to put together and validate are of special interest to our utility partners. Next slide. So some results from that first study at TUMWRF. So what we're showing here is average DAWN removals for the 3 coagulants that we evaluated. So alum in green, polyaluminum chloride in blue, and ferric chloride in orange. So the feed for all of these coagulation tests was effluent from our denitrification system at TUMWRF. So our starting DAWN levels are already quite low, somewhere in that 1 to 3, sometimes 1 to 4 milligrams per liter range. So we're starting with a very low concentration feeding into these coagulation studies. Also shown there on the figure along the bottom, you can see we've got a pretty robust sample set with our number of samples in blue. And you can also see that a few of our tests evaluated the use of an additional polymer. Although we did get some better results, our utility partner wasn't interested in that. So ultimately, we didn't pursue the polymer. But what we found was that enhanced coagulation was capable of pulling off a lot of the DAWN. We see 40% or better removal of DAWN with our blue and our orange columns there. So the ferric chloride performed a little better than the PCL, but it did require much higher doses to give you equivalent removal. Bigger picture, this work validated that the enhanced coagulation approach is a viable treatment option for facilities that are facing those ultra-low nitrogen discharge limits, especially where that recalcitrant DAWN fraction is going to be a major portion of that last little chunk we've got to remove. So next slide. So the next project I want to briefly discuss is that pilot scale demonstration in collaboration again with our local utilities and in this case our local city government. And it's part of the One Water Nevada IPR demonstration. So the project was evaluating advanced water purification for groundwater recharge. So DAWN was only a small portion of that overall research effort. We just had a couple of months, 2 months operating window there where we could dedicate it to DAWN. But for all the reasons that we've already discussed, nitrogen removal is a major concern for our region. So our partners really jumped on the opportunity to include that in their technology assessment. It also gave us a chance to evaluate the potential for nitrogenated DBPs to form, so those hands and hams that I showed on the previous slide, in that advanced treatment train. So next slide. So this one's another result slide. I apologize, it's fairly busy. I consolidated several into one. Also apologize, I removed the error bars just to make sure that we could see the trends. So what we're looking at here is concentrations through the treatment train of our dissolved organic carbon. So those are the brown skinny bars. And then our different nitrogen species, those are the fatter stacked bars. And I've highlighted DAWN there in green. The percentage values that you see there in black for the carbon and in red for DAWN correspond to a stepwise reduction from each step in the treatment train. So as you would expect, the DOC concentrations decrease throughout the process, and we see the greatest reduction coming in our BAC and our GAC steps. From a nitrogen perspective, we can see that the ammonia, the little purple bars at the top, is essentially a non-factor. And then, as expected, the nitrate in the gray, the bigger parts of the nitrogen bars, are fairly constant throughout that advanced water treatment train. But what we do see is a pretty good reduction in our DAWN concentration, so the green bars or the green subsection of the fatter bars throughout that treatment train. And our biggest changes again happen with our BAC and our GAC steps, which is what we would expect. Dissolved organic nitrogen should be removed when we're removing that organic carbon as well. So next slide. If we look at that same data, but if we kind of change our focus only to be on DAWN and then overall DOC, and we look at the removal, again, on that step-by-step basis through the treatment train, we see reasonable correlation between the removal of the two species, with big gains, or I guess rather big losses, for both groups at the BAC and GAC steps. However, we also are able to see some selective DAWN removal over carbon removal at the coagulation stage. That's highlighted with the blue circle there. So this data set, or looking at the data set a little bit differently, tells us that we do have an opportunity early in our treatment train to selectively reduce DAWN, if that's our target. And one of the benefits of having that early in the treatment train is by pulling those organic nitrogen levels down early, then we reduce the potential for the nitrogenated disinfection byproduct formation. So as part of advanced water treatment processes or trains, this is important. So speaking of those advanced treatment trains, so this is a slide I'm pretty excited to share. This is our artist rendering of the advanced water purification facility that's coming to the Reno area soon. This is a culmination of a lot of effort by a lot of different people, including the work I just showed you. And it's actually the first potable reuse project in Nevada. So just earlier this month, all of the permitting was approved and paperwork done. So we're expecting to break ground. We're hoping for late June, but maybe early July before we get there. So this facility is going to be a 2 MGD, 2 million gallons per day facility, adjacent to that Reno STED WRF. So it's going to take about half of the STED effluent and treat it for groundwater recharge. And it's going to be going into a basin north of Reno. It's very well characterized, very well monitored. And the first phase of that on the withdrawal, on the pumping, is going to be for agricultural reuse, so we can really monitor and understand what's happening out there in that basin. And then eventually the goal is for that to become a supplemental drinking water source for us. But probably the coolest part of the facility, or at least from my perspective, is that our partners have dedicated about 1,800 square feet of dedicated research space just for the university. So we're going to have our own space there, all plugged into instrumentation and all flows, where we can continue some of this great collaborative work that we have with our utility and our government partners. So stay tuned, hopefully, for more great things to come from that.
Nice segue, Sage. Speaking of collaborations, we're going to wrap up with our collaboration slides. So we are RAP 7.10, so that's US EPA's RAP, and RAP 2.0 that we work closely with, and of course with NAWI, as you've heard. We've listed most of our utility and government partnerships, as well as our military partnerships here. And I just want to wrap, I don't know if Peter's still on, but hey, I had to wrap up with this image of him. This was Industry and Academic Partnerships for Improved Pilot and Demonstration Testing. It was a session that we had at the 2026 Water Reuse Symposium. Good. So we wanted to save time for Q&A.
Yeah, thank you, Amy, and everybody. That was fantastic. Thank you for giving us the overview of what a rich and dynamic program. And I see both some common themes with NAWI, including the idea of small scale package systems, and Andrea, that idea of electrification and making these systems run just with electrons. That's fantastic. As people put in questions, I had a couple. Andrea, can you go back to your section, or maybe Amy? I think it was slide 32.
Okay.
It's the one where, let's see.
Unexpected.
Let's see if we can add.
32. That's right.
32. Yeah, so I had a little question. So am I interpreting this right? The silica goes up from the FBCR to the iron reactor. Is that true? And if so, why would that happen?
That is what. Yes, and that's in this case is because of dilution. Basically, we do not remove. So we don't remove any silica from in the ion reactor. If we go back to the schematic,
Yeah.
Amy,
I should have.
All right.
I should have. I have it here.
This one?
yeah,
Hey,
one
we
more.
started delving into first the
Two more.
Amy conversation. That,
Yeah.
There.
So basically,
yeah,
there is the
Julie had
mixing
earlier,
point.
something like that
So
went really
what we
well.
measure
People,
after the
around
ion
globe,
reactor is
Mhm.
everything basically.
of all
So
places,
and from the
sure,
way we
but-
run the experiment from the electrolytic cell on the acid there, there is some silica going on there.
Okay, that's interesting. And Andrea, I don't know what you know about NAWI Project 3.17, but we're using a process called virtual curtain, which was a precipitation technology developed by CSIRO in Australia. Jim Lozier and Jacobs piloted this at Yuma, Arizona. And
Okay.
we're getting very high silica removal rates. And that gets into my second question is, Andrea, 90% water recovery from concentrate, great job. What's limiting it? What's limiting it at this point?
The salinity, the sodium chloride, the salinity of basically of the inner cycle that we have. Because this process, basically, you should think about it as a closed cycle, which the secondary arrow concentrate produces the acid and base and then it puts back together, right? And so inside the sodium chloride will increase until we bleed it. And so we have essentially a seawater desal there. So it's 1,000 psi, 70
So,
grams
but you
per
could conceivably, you could then take that to some high pressure RO or some,
Oh yeah,
brine,
Yeah.
concentrated brine production.
yeah.
Very
This
good.
is definitely a pre-treatment process. It's not married to RO. We can have it with membrane distillation and other technologies for
Very
separation.
good. Very good. Excellent. So back to you. I had a question for Adam. Adam, I will admit, I'm not, this is not my space, and so antibiotic resistant genes as essentially a pathogen. I didn't realize that could be transferred. So can you tell us a little bit more? I mean, we don't regulate for that in standard wastewater treatment. And is this a factor in water treatment that we should be paying closer attention to? Passing of antibiotic resistant.
Yeah, so these resistance genes spread via what's called horizontal gene transfer. So they can spread in a few different mechanisms, and it's very difficult to track in highly complex microbial communities like what we have in our wastewater treatment plants. So this is an example of using a new advanced molecular biology tool to get a better sense of how permissive these resistance genes are on these mobile plasmids. But we need to do better additional work to understand like rates of transfer, et cetera, or what different conditions lead to increases in the transfer. So we're just kind of at the tip of the iceberg in terms of redesigning or operating our systems differently to reduce antibiotic resistance.
Yeah, but it looks like the anaerobic MBR seems to be clearly better. So
yeah.
One step.
Mhm.
Good. Okay, very good. Nick, are you still there?
Yes, I am.
Yeah, so so this is an exciting consortium and a lot of exciting work. I want to have you circle back to some of the insights that you're gaining and implications for Department of War facilities and installations. So are we going to see our facilities and installations start adopting water reuse? And also, are there any current facilities or installations that are already like kind of starting to build in this infrastructure? And what are we learning from that?
Right. So I'm part of the research arm of the Army Corps of Engineers. So we hope, yes, that there's widespread adoption. That's the reason why we do these pilot programs and things like that. So there are examples. There are installations that have adopted a few purple pipes, using reclaimed water for watering the grassy areas and things like that.
We know the
Plants.
generals love their green golf courses.
Yes. Yep. And for some car washing, there's some really good research being done in the forward operating areas with containerized systems, shower systems that can reclaim the water, reuse that gray water from the showers. There are a number of, mostly being used in not widespread, but we are starting to get penetration because thankfully people are acknowledging that this is an issue that is not going to go away, that we really need to address it, and we can't keep doing what we did in the old days.
Right.
So there's a number of nice containerized systems for the forward operating base, either capturing showers, different gray water reclamation, and then at our fixed installations, we are looking at what else can we do with the many of our installations do have wastewater treatment plants, facilities on site. So how can we upgrade those? How can we modernize those to do it? Now, the tricky thing is, as you know, there is no national standards. Every state, even down to the regions or localities, have rules on what we can do. And there are some states that until recently you couldn't even put rain barrels out in some areas, right?
Colorado, we're talking about you.
Yeah, some unnamed location, but these are very interesting. So because our installations are in multiple different states, we have to be concerned about what is downstream from us, and there is a big push. So it's very, very good to see that we're getting starting to see some momentum. Great question, Peter.
Yeah, well, and I think the other thing that's interesting about the Department of War is, first of all, you're the largest building owner on planet Earth. I think you have almost 1 million buildings across the globe in terms of the built infrastructure. It's really, really, and it represents an economy that when you all decide to do something, you can implement change very rapidly, as opposed to we know, how slow in the municipal sector, how slow it is to adoption. So I'd say given that, and given that the Department of War can be a very fertile and kind of active partner, what else would you like to see from the research community? Like for the people who are tuning in from around the country, do you think that they should be talking to their base commanders on how do you recommend researchers engage with the Department of War and its facilities if they want to explore water efficiency or some of these other things?
Yeah. So one of the best ways is keep your good ideas coming. I think you will see over the next few years, you'll see funding opportunities, proposal opportunities, that are out there. And we want your good ideas. When those requests for proposals come up, we want you to do that. Make sure you get your good ideas in there. Of course, as private citizens, you should always be reaching out to your both state and federal representatives. Always a good opportunity. If you have a concern, if you want to see that, again, that's our right as citizens. Always, our representatives, that actually makes change when they get, from their constituents, if they get information or requests or things can actually happen when you do that. I think there's these advisory panels, participating in these, public-private partnership opportunities. If you can come to and attend those events and keep this conversation going, all of that helps build the momentum. And we're seeing, like I said, we're starting to see people acknowledge this, and it's a very good research area.
Another thing, a number of people in the NAWI community, researchers in our community, are active participants in CERTUP and ESTCP. These are two programs out of the Office of the Secretary of Defense that focuses on environmental stuff. Can you tell me a little bit, or either suggestions, or is there explicit interrelationships between ERDIC and these programs, or just how do you see that?
Yeah, the ESTCP, those OECIF, OEPF, you might hear those are acronyms of different funding opportunities. So, and many of those come out yearly, and there are topic areas that have research requests. So, like our laboratory and our laboratory system, which is the ERDIC, we can participate and jointly write proposals. We can also help facilitate, some of those funding opportunities need to have a demonstration at a military installation. So historically, ourselves and some of the other DOW laboratories can be partners on those. There's some paperwork to get in place. We want to do it all appropriately because we want fair competition and all. But through our laboratory system, or actually contacting the ESTCP program themselves and say, hey, I have this technology, or any of those funding opportunities, who could I partner with to help me find a facility to do this demonstration? Because a company or a university might not know how to go about reaching that that base energy manager.
I would say almost always we have no idea how to reach that base commander.
And the people who run those funding programs are excellent about connecting us, and they'll reach back to our laboratory and other laboratories. Or if you contact our laboratory directly, we want to help because we are researchers. We want to make sure all appropriately get you connected with people to make these proposals more successful. So don't be afraid if there's a point of contact on those. Take advantage of that. If there's a webinar, the people running those demo programs, they want to see success. They want to see the best proposals possible also. So don't be afraid to reach out to us.
Very good. We know where to find you, Nick, that's for sure.
Yep.
Amy, I'm going to turn back to you as kind of the final kind of question, but also, integrating thoughts that you have.
I hadn't heard about that.
This program, tell us, first of all, how much longer does the collaboration exist? One, and two, what do you all hope to be the kind of theory, what the change that occurs after you all have finished your work? Don't forget to unmute yourself.
Sorry. We were initially 4 years. Probably that's going to easily be 5. So that's where we're at now. We will see if we can extend that longer. I think a real space that we're targeting is not that real future. Well, there's some more futuristic research, but really applied research that fixes our problems. Although we think of in like potable reuse as being there and done, it's not yet. And there's a space in here that I think academia is needed and these collaborations are needed to fill. So I think that's kind of our target research area. But we also feel like we have access to people who can do some of this community outreach, community assessment, and other aspects. You asked a second question too. What did I not answer?
I just said, I think that this collaboration comes to a close, but it's not the end. And so, and so I would say, like, one of the things I hope to see, Nick, is in 5 years' time, it would be great for us to have some sort of advanced reuse demonstrator going on at one or more bases or facilities. I think it'd make a big difference. And I'm not putting a fine point on it. Generals like green grass on their golf courses, so nothing would excite people to a beautiful, sustainable golf in somewhere on a base. So everybody, on behalf of NAWI, thank you again so much for joining us today. And everybody who's listening today and listening to the recording, our next webinar is on June 17th. And you can find this and other recordings on the NAWI YouTube site. And for all of us here at NAWI, we want to thank you all. And please reach out to the speakers today if you have more questions. And we look forward to seeing you on future episodes. Amy and team, thank you all very much. Take care.
Thank you,
Bye-bye.
Peter.
Thank you.
Thank you.
Three little grandkids, both